From a single fertilized cell, a magnificent symphony of life unfolds. This is the incredible journey of embryonic development. One of the earliest events in this process is cleavage, a series of rapid cell divisions transforming the zygote into a multicellular marvel. Imagine a sculptor meticulously carving a block of marble, each strike revealing the intricate form within. Cleavage is the sculptor's chisel shaping the raw potential of the zygote into the foundations of a complex organism. These divisions are not merely about increasing cell number. They lay down the blueprint for the future embryo, dictating the fate of different cell lineages. The driving force behind cleavage is a delicate dance between the cell's nucleus and its cytoplasm. The nucleus, harboring the genetic blueprint, directs the division process. Meanwhile, the cytoplasm, a rich soup of molecules and nutrients, provides the building blocks and energy for these divisions. However, not all eggs are created equal. The distribution of yolk, the nourishing substance that sustains the developing embryo, varies dramatically between species. This variation in yolk distribution has profound implications for the pattern of cleavage. Think of an egg as a cosmic cradle, nurturing life in its earliest stages. The yolk, a treasure trove of proteins, lipids, and carbohydrates, serves as the primary energy source for the developing embryo. Based on the amount and distribution of yolk, eggs can be broadly classified into 4 types. First, we have the isolecithal eggs, characterized by a small amount of the yolk evenly distributed throughout the cytoplasm. These eggs are like well-provisioned spaceships, ensuring a uniform distribution of resources. Next are the mesolecithal eggs, containing a moderate amount of yolk concentrated towards one pole, known as the vegetal pole. This creates an asymmetry in the egg, like a planet with distinct hemispheres. Telolecithal eggs take yolk concentration to the extreme, with the vast majority of yolk packed at the vegetal pole. Imagine a giant yolk sac tethered to a tiny island of cytoplasm. Lastly, we have the centrolecithal eggs, where the yolk occupies the central region, surrounded by a thin layer of cytoplasm. This configuration resembles a celestial body with a molten core. Each of these yolk distribution patterns dictates a specific cleavage pattern, shaping the early embryo's form. The dance between the dividing nucleus and the yolk-laden cytoplasm gives rise to fascinating variations in cleavage patterns. In isolecithal eggs, such as those of sea urchins and humans, cleavage furrows effortlessly bisect the egg completely, resulting in cells of roughly equal size. This type of cleavage is known as holoblastic cleavage. Mesolecithal eggs, like those of amphibians, present a slight obstacle course for the cleavage furrow. Due to the concentrated yolk at the vegetal pole, divisions are slower in this region. The result is a pattern of unequal cell division, with smaller cells at the animal pole and larger, yolk-rich cells at the vegetal pole. This is also a type of holoblastic cleavage, though modified by yolk distribution. Telolecithal eggs, exemplified by birds and reptiles, pose a formidable challenge. The massive yolk mass restricts cleavage to a small disc of cytoplasm at the animal pole. This incomplete cleavage, known as meroblastic cleavage, results in a disc of embryonic cells perched atop the yolk. Finally, centrolecithal eggs, commonly found in insects, exhibit a unique pattern called superficial cleavage. Nuclear divisions occur without cytoplasmic division, creating a syncytium, a single cell with multiple nuclei. Later, the nuclei migrate to the periphery, where cell membranes eventually form, and the distribution of yolk, a seemingly simple factor, exerts a profound influence on the earliest stages of embryonic development. It dictates the pattern of cleavage, shaping the size and arrangement of cells in the early embryo. This, in turn, influences cell fate and the establishment of body axes. For instance, the unequal cell divisions in mesolecithal eggs lead to the formation of distinct germ layers, laying the foundation for specialized tissues and organs. The diversity of cleavage patterns observed across the animal kingdom highlights the remarkable plasticity of early development. It underscores how evolution has sculpted a fundamental process to accommodate the unique challenges and opportunities presented by varying yolk distributions. This interplay between yolk and cleavage is a testament to the elegance and efficiency of life's solutions, shaping the destiny of countless species from the very first moments of their existence.